Wanted to welcome everybody to the Cellular Dynamics workshop on large-scale production of human iPS cell-derived cardiomyocytes. My name is Chris Kendrick Parker. I'm the Chief Commercial Officer of Cellular Dynamics. Other participants in the presentation will be my esteemed colleague, Blake Anson, who is a study director for our HERG screening lab at CDI, as well as the product manager for our human iPS-derived cardiomyocytes. We'll break the presentation up into two parts. One is really kind of giving the overview of where we are as a company and who we are and what we're trying to accomplish. And then Blake will give a great deal of detail around the types of cells that we're making currently, and especially the cardiomyocytes. So we'll give you kind of an understanding of the founders and our current operations and where we really see the distinction and the power of using inducible pluripotent stem cells as a starting material as opposed to embryonic stem cells. We'll go through what we think is really critical to deliver a viable tool, which is give you an idea of our manufacturing process, because we are truly manufacturing these cells in large quantities to make them useful as a cellular tool for screening. Give you an idea of what we're trying to do as far as QA and QC, and then the general process and purification that hopefully will give you confidence that what we can deliver is indeed what we say. And then I'll turn it over to Blake, and he'll get more detailed information and data around the characterization of what we call I-cell cardiomyocytes, being that they are derived from inducible pluripotent stem cells, both on the gene and protein expression, as well as the electrophysiology and cytotoxicity applications that many of our current collaborators are utilizing today. And then we'll have some conclusions and then entertain any questions that you might have. So the company, CDI, was founded out of the University of Wisconsin-Madison by Jamie Thompson, who is a pioneer in embryonic stem cell, was the first to derive human embryonic stem cells, also was the first to, along with Shinya Yamanaka out of Kyoto University, the first to derive human inducible pluripotent stem cells, and then most recently in April of this past year was able to derive human induced pluripotent stem cells without the integration of vectors or DNA, so basically vector-free or footprint-free iPS cells. These are really what's going to be critical for any long-term therapeutic applications from an autologous therapy perspective. We have capabilities for both with CDI. The other founders were really amalgamation of collaborations that occurred with Dr. Thompson. Dr. Thompson's work was really to create stem cells and keep them as stem cells in their pluripotent state. But he realized that in order to make them have utility, really the value comes when you can differentiate and control those cells and differentiate them into terminal cell types that elicit the same human phenotype as the individual that they come from. And so he engaged in collaborations on the hematopoietic side with Igor Slukvin, who's also a founder of the company, and Tim Camp and Craig January on the cardiovascular side. Craig January being the person that derived the first HERG line that's used primarily for screening and IND filings for ion channel and ion manipulation. And Tim Camp is also a cardiologist that really worked on the differentiation protocols with Dr. Thompson, and many of those things were converted over to the company. With this team of founders, we've been able to recruit a large number of human stem cell biologists within the company. We did some math recently in terms of what we have on hand at this point. We currently have 70 employees at CDI, over 50 of which are actually research and development scientists that have human stem cell experience. We currently, even though the field is only about 11 years old, we did some math, and we have over 130 years of cumulative human stem cell and differentiation experience within the company, broken down into a variety of different areas, notably in the creation and derivation of iPS cells from a variety of different individuals. We have recently developed a method that we can derive human iPS cells from standard blood draw. So we no longer need a hair follicle or the use of a skin punch biopsy or fibroblast to derive, which really puts it into a clinical arena that can be used in standard clinical practice. And then we also have the different cell biology groups. The company is primarily broken down into process science, product development, research, and IPS technology. Junying Yu is a person that recently joined the company. She was the first author on both of the IPS papers recently, both the vector-containing and the vector-free that were published in Science in end of 2007 in April. And she pretty much, as a postdoc coming out of Jamie's lab, could have gone anywhere she wanted, and she decided to stay in Madison and come to the company, and is really bringing her methodology and expertise into CDI. We're very happy to have her leading these groups. So the concept here is that we switched recently from use of embryonic stem cells to almost exclusively manufacturing, production, and engineering with induced pluripotent stem cells. And the concept here is that from a blood draw or a skin biopsy or even a hair follicle pluck from keratinocytes, we can, by the introduction of some genetic material, reprogram these cells back to a stem cell state where they have full pluripotency and the ability to differentiate into any cell type in the body. These are, for all practical and all measurable purposes, identical to human embryonic stem cells without the political and ethical constraints that come with that arena. And we've, by practical measures in terms of the differentiation methods, in terms of taking them and making any cell type, we haven't found any differences in terms of the ability to take these iPS cells and make them into any cell type in the body, any of the 200 cell types. What this does in terms of conceptually, it kind of changes the game in terms of the use of a cellular tool. Instead of having a generic model that is produced into a cell type of interest, we now have the ability to represent individual biology. We can basically recapitulate what happens in a clinical trial in a petri dish. We can make iPS cells from virtually anyone, and we can take those cells through the differentiation process and produce the terminal cell types of interest. You'll hear our first entree into that is human cardiomyocytes, but this now runs the gamut of taking us away from just kind of a toxicology normal cell tool to a tool that can be used in preclinical and clinical research. We're in the process right now of attaining samples from individuals with appropriate informed consent such that we can make an ethnic diversity panel. We plan on having about 5 different ethnicities represented in that and having individuals in a statistically relevant set that can be supplied either as groups or as an entire panel. In essence, creating an in vitro clinical trial. We also can look at genetic predisposed individuals. We're also trying to generate samples with our academic collaborators to represent genetic long QT syndrome by producing iPS-derived cardiomyocytes from those individuals, as well as looking at hypo and hyper responders to chemical entities so that we can represent the full gamut that's seen by a molecule as it enters the clinic. And then in addition, these tools, even though they're normal cells, have a great deal of propensity for use in discovery biology as well. In addition, we have this retrospective analysis. We're working with a pharma company right now to potentially take idiosyncratic events or individuals retrospectively in clinical trials, primarily phase 2, that have elicited a strange EKG, for instance, and we can now retrospectively take a sample from those individuals, derive their iPS cells, and make their cardiomyocytes such that we can really understand the mechanism of toxicity of those individuals. We think that this is really going to train the pharmaceutical industry much more effectively to provide better medicines that don't have these idiosyncratic events in clinical trials. So the whole concept of this is that there's a lot of cool science that happens in stem cell biology in terms of people making a variety of different cell types, but unless you can produce sufficient and significant quantities of these cells, not only do they not have utility in a research mode, but they won't have any utility in a cell therapeutic arena as well. It's going to be critical to then put those into a situation where we have the ability to move towards a quality measurement system, GLP or GMP and ISO-related systems such that we can truly manufacture these in a controlled manner. I'll take you through this process, and we're actually developing systems right now to do this. And the interesting thing is we can't exclusively use ISO or GMP to do this. We have to kind of marry those two disciplines to actually come up with something that will be useful in this arena, because in ISO, for instance, it's much more related to customer satisfaction. It's really around the entire organization, and it's systems related. Whereas since we're producing a product, we also need to incorporate customer protection from the GMP perspective, manufacturing of QA and QC, and shipping and receiving, and then it's much more product related. So none of them fit our needs completely, but we're basically moving towards an amalgamation of the two, because this is critical when it relates to the materials that are being used, not only from the disciplines of the manufacturing process, but also in terms of managing materials. We have to incorporate, compliance and product consistency, calibration of the various instruments that we're using. Really, one of the key elements is the supplier quality and management. We're actually purchasing materials for this differentiation process from manufacturers that have never produced the volumes that we require. And to manage that from a source and second source to make sure that when we produce these cells, they're consistent and reliable and that you can use them in a long-term longitudinal manner, this is really one of the critical steps to get us to that point. And we also have the ability for complaint handling in terms of being batch record analysis and quality control. The manufacturing process is really broken down into 3 phases. Right? The first is to make billions of stem cells as a starting material. So you should think about the iPS cells as the starting material that's used to differentiate into the terminal cell types. You have to produce significant quantities of these cells as a starting material because most of the processes of differentiation are not expansion related. You need more starting material going in than you get out at the end. In the research mode, most people that have been trying to produce cardiomyocytes, for instance, the yields that they've generally gotten have been in the low single digits, 1% to 2%. In terms of generating pure cardiomyocytes. We've now pushed this by the methods that I'll describe up into the 20 to 30% range such that we can make it cost effective and produce significant quantities. So we're actually producing more iPS cells on a daily basis than we believe anybody in the world is. We're producing 1 billion iPS cells a day as starting materials that goes into the differentiation protocol. The differentiation protocol then is a process of adding cytokines and cocktail of reagents that cause the cells to directly differentiate into the mesoderm lineage and then into the cardiomyocyte terminal cell type. And the way we've done that is a patented, protected, proprietary process where we genetically engineer the iPS cells initially to have a pan-cardiac specific promoter with an RFP or a fluorescent protein cassette that was really useful in optimizing the process, and then a antibiotic resistant component at the end that allows you to then purify the cells once you produce them. The fluorescent protein modality that was used was really influential in optimizing the process and making us understand how we could get the yields up. And the reason I say that is because the only way that you know when you look at a cell population that you've created cardiomyocytes in that population is when they start to spontaneously beat. So if you're trying to produce a large number of those cells and you're trying to optimize the process, and every time you have to do that, you have to look into the microscope and look for more beating cells than you had the last time, it's not a very efficient process to optimize. With the RFP, as soon as we saw glowing cells and we could measure that through FACS sorting, we started to understand how we could push the yields up by the manipulation of the process. And then, in addition, we have this resistance gene that we can apply. Once we've generated large numbers of cells, we can apply an antibiotic that basically eliminates all the other cell populations and provides a highly pure cardiomyocyte population. And I can take you through some of that data initially. But really what this has done and what we've spent a lot of money and a lot of effort on within the company is to produce a pipeline that we can now start to put other cell types through with this same process. Cardiomyocytes was our first entry based on our expertise and the unmet need that we see in the market. But really, this pipeline can be applied to hepatocytes, neural cells, chondrocytes, osteoblasts, any cell type that could have potential utility in a screening and biological testing mode. And so when we produce these cells, we have a pre-purification process where you see a large number of aggregates that are clumps of cardiomyocytes that are grown in suspension. You can see when you look at them under fluorescent conditions, a percentage of those are glowing red, thereby indicating the percentage of cardiomyogenesis. In the case of the pre-selection, you see about 13 or 14% cardiomyocytes in that population. And then post-purification, after we apply antibiotics, you see a much greater propensity of red glowing cells, and in this case, you see a representative 98.05% cardiomyocytes in that population. This is generally within the specification that we see on a routine basis now. So I should also say that because it's a pan-cardiac promoter, the population of cells that were in this set are both atrial, nodal, and ventricular. They're about 50% ventricular cells, such that when they're plated down onto a monolayer and allowed to create connections, they will beat spontaneously individually, but when they create a syncytium across the well, they will beat synchronously across the whole field of cells in the dish. And here's a representative of that in terms of seeing the actual beating cells. These are aggregates of cardiomyocytes that are probably in the hundreds or thousands per aggregate of cardiomyocytes, and you can see them actually spontaneously beating. This is kind of the science fair slide that once I put this one up that I could basically say goo goo and gaga, and no one really would care. There's nothing like looking into a microscope and seeing cells that are spontaneously beating and wonder how that could possibly occur. These are the human cardiomyocytes. They're the current product that we're launching in Q4 of this year from a full commercial standpoint is RFP labeled, so these are red glowing cells. They're all iPS derived, so there's no embryonic stem cell material here. Between 98 and 99.X% pure. They come with a certificate of analysis that determines exactly on a batch per batch basis what purity they have. They have the antibiotic resistance, and these cells will actually, once plated down, beat in culture for significant periods of time. We've had cells in the laboratory beating in culture for months. Literally months. So you could potentially do longitudinal chronic exposure types of studies with these cells. And these are going to be available for full commercial launch in Q4 of 2009. I will also say that because we have a great degree of our customers that want to use high content imaging with these cells, that we're going to a colorless version of these cells now that the process has been optimized, such that the red channel will also be able to be used in a multiplex manner for high content imaging. And then we'll also have, in the future, potentially have inducible promoted cardiomyocytes, such that you could induce a color change if in an assay component. So with that, I'll turn it over to Blake, who will give the information on the characterization and the measurements of these cells and the applications.
All right. Thanks, Chris. So we're going to go through a cardiomyocyte, just a characterization. This will be looking at genes, proteins, and their EPhys. And the way we've set this up is it's going to be a basic characterization, and then we're going to kind of look at applications that you may use back in your shop, and that will also then provide some further characterization data. So the first is to look at the gene expression. Here we see a panel of qT-PCR experiments, so real-time quantitative PCR. Relative expression is on the y-axis, and on the x-axis, you've got days in our induction process. Day 0 is when they're still stem cells, and they're just about to enter into our differentiation process. And day 32 here is out at about release age. Here we see a stem cell marker, OCT4, that goes down, which is what you'd expect. These are no longer stem cells once they enter into the differentiation process. They're on their way to becoming cardiomyocytes. The other two transcription factors we see here are NKX2.5, which is a general cardiac transcription factor, and the IRX4, which is a ventricular-specific transcription factor. As you'd expect, they go up over time. The other genes down here are various cytoskeletal elements, actin, myosin light chain, myosin heavy chain, and your troponins. And the troponin I here is our marker of maturity. Again, these all come up over time as one would expect. Second set of panel here are the ion channels. Again, we've got sodium channels, calcium channel, HERG, or potassium channel, and then the KCNQ1, this is the IKS current, and its subunit MIN-K, KCNE1. And again, these all come up over time, reaching measurable levels at the release age. Second would be just a very brief protein expression. Here we just have a field, looking at phase or bright field of cardiomyocytes that's been laying down. And here there's just the NKX2.5 stain, saying that we've got a lot of cardiomyocytes. And here we see the cytoskeletal organization, the sarcomeric organization. We've got actin, and we've got the nice striated phenotype that we'd expect, as well as a cardiac troponin. And if you use your imagination, you can see the striations here as well. So they show the expected pattern of distribution. Second would be the action potentials that we see. In the adult human heart, you'll see different action potential phenotypes from the different regions of the heart. So, for example, in the nodes, you've got these beating, more rounded action potentials, versus down in the ventricular, you've got these potentials that are longer in duration. Here we see 3 of the primary action potential phenotypes recapitulated in the stem cell-derived cardiomyocytes as well, where we've got an atrial-like, nodal-like with a prominent phase 4 depolarization, and a ventricular-like with a prolonged action potential duration. Now we say these are all atrial-like, nodal-like, or ventricular-like because they do spontaneously beat. That's both the benefit and the curse of the stem cell-derived cardiomyocytes. The benefit is they're spontaneously active in culture. They've got a high metabolism going, so you're really able to test your drugs as if they would be in vivo. The downside is, well, they beat, and your ventricular cells by themselves do not beat. So one of the ongoing projects that we have at CDI is to further this maturation process. But right now, nobody can do that, and this is really the state-of-the-art technology at the current point. All right, so next, I'd like to look at the applications of these cells. We're going to start with the ion channel electrophysiology, going through the various patch clamp type recordings, and as well as an assay known as a microelectrode array recording, and then some of the cell-based cytotoxicity assays, looking at general in viability, cell death, ATP, and mitochondrial function. As we go through this, we're going to try to give just a really rough outline of the steps and the timelines of what you'd have to go through in your shop so you can kind of get a feel of how you'd work with these cells actually. First is the shipping and the receipt. These are going to be cryopreserved single-cell suspensions. Those come in simple cryopreservation vials. Each vial will contain enough cardiomyocytes to populate either 1 or up to 5 96-well plates. This will be a catalog item that you can then order based on your specific needs. We'll ship them on dry ice, and then the long-term storage would be in liquid nitrogen, and they'd be ready to thaw, use, and plate as you'd see fit. So for conventional patch clamp recordings, the basic steps here would be to, day 1, thaw and plate your cardiomyocytes onto coated cover slips. These coatings can be with either gelatin, fibronectin, collagen, laminin. We haven't noticed significant differences between the coating substrates. We allow them to just recover under standard culture conditions for at least 48 hours, and then transfer that cover slip onto the stage of your conventional patch clamp and start recording. And so now we're going to go through a series of slides showing the currents that we've recorded. First here is the sodium channel current. This is responsible for the upstroke of the action potential. It's a very rapid and fairly large current that's present in these cells. So here we see a family of currents showing that they're activated with voltage depolarizing steps as shown here. If you plot voltage versus the current, you see the inward current that reaches its peak around minus 25 millivolts and then begins to rectify. And just a normalized conductance graph shows that you've got a V1 half at about minus 40 millivolts with a slope of about 6 millivolts per E-fold change. This is well within the realm of published values, and so these channels are behaving normally. Secondly, we would look at calcium channel currents. These are responsible, if you look at the action potential, these are responsible primarily for maintaining that plateau. They are smaller. The way we've illustrated them here is we go through a family of depolarizing steps, as illustrated down here. Then we apply a calcium channel blocker to block all the calcium channel current, apply that same depolarizing steps, and then subtract after from before to get your calcium current. Again, we see activation through the series of steps that then begins to rectify. If we plot this as membrane potential versus current, we see a nice, again, inward bell-shaped peak that rectifies. We've got a peak of around 0 to plus 5 millivolts. We do a conductance plot, again, fit with the Boltzmann distribution, and we see a V1 half of about minus 8 millivolts. The last current we'll see from the patch clamp here is the current that probably most of you in safety are familiar with, and this is the herd current. This is the current that is the bane of many manufactured drugs. In endogenous cells, hERG has a very low expression. But even though it's got that small expression, when you block it, you can get that prolonged action potential. So the way we see hERG current here is, again, through a pharmacological subtraction protocol, where we have our control traces and wash, then we add a completely blocking concentration of a drug, do, again, repeat the voltage protocol, and then subtract after from before. And here we see the hERG current with its telltale tail current right there. If you plot IV, you see the standard bell-shaped curve of the current during the step with a peak here around minus 20 millivolts. And if you plot the peak tail current as a function of voltage, you see its sigmoidal properties, sigmoidal shape, with a V1 half of about minus 27 millivolts. This is classic signature of hERG current. So we're seeing that these cells, through your conventional patch clamp, you can fairly easily measure both sodium, calcium, and hERG currents. If you're looking at current block and how it affects the action potential, The conventional patch clamp is also one way to go there. And here is just an example of what an experiment may look like. You've got a long train of action potentials up top here. It's at fairly low temporal resolution. You've got basal conditions. You add a physiological relevant concentration of a hERG channel blocker, wash out, and then add calcium channel blocker, and again wash out. Down here, we see expanded action potentials. Under basal conditions, we've got our normal ventricular action potential. Nice wide base, repeating rhythmically. Under E4031, which blocks hERG channels, we're going to start to see that action potential prolong and even start to trigger what's called an early after depolarization, or an EAD. This is commonly thought to be an arrhythmogenic trigger. These can become quite pronounced, and under wash, you get your action potentials returning back to near normal conditions. You add nifedipine, which blocks the calcium channels, and there we see the spontaneously beating goes away. Calcium channels play a large role in that spontaneously beating aspect. When you wash out, you get a return of the action potentials again. So it's a very powerful tool to record the effects of your drugs on ion channels in their native or near native environment. And again, just to summarize, you have measurable differences in your action potentials, both in the depolarizing currents. Here we've got calcium. Looking at nifedipine, we've got a concentration-dependent decrease in action potential duration. Obviously, these concentrations are below what would cause the action potential to stop firing, as well as potassium, or the hERG IKR block. Again, you see a dose-dependent or a physiologically relevant concentration of E4031 producing a very prolonged action potential. The other common way of looking at the electrophysiology of these cells is the automated patch clamp. And with endogenous, or with native cells, native tissue cells, this has been quite a hurdle to overcome, because these cells, the automated patch clamp likes nice round cells. Muscle cells are typically not nice and round. The stem cell-derived cardiomyocytes, once you bring them up from the plate, are more amenable to the automated patch clamp type experiments. And so here again, you'd thaw and you'd plate your cardiomyocytes, allow them to recover for 2 days or more. Once they've recovered a bit, you see them beating and they're active, you can go ahead and trypsinize them again, and then just keep them in a vial in suspension and add the aliquots to the automated patch clamp. These initial preliminary investigations were done with one of our collaborators, that's public against Roche, out of Palo Alto, and now moved over to Nutley. Here we just see a couple of example recordings. We have the family of sodium currents, sodium channels, and when we plot current versus voltage, we see that inward sodium current with the rectification. Again, we're seeing our peak inward current at about minus 25 millivolts. Calcium channel, it's smaller, but you're able to record it on the automated patch clamp, and again, we see this inward rectifying shape around here, minus 10 to 0 millivolts. So in the automated patch clamp, we've readily been able to record sodium and calcium currents. Our success rates can reach greater than 50%, and this is just with initial experiments. With a bit of tweaking and messing around, I believe that these success rates can reach that obtainable with the typical transfected cancer cell lines. And this will really allows the automated analysis of ion channels in an endogenous system. All right, the last type of ePhys data that I'll be going over is called the microarray recording. And again, I'm going to stop, take a little side note after this slide, I believe, and kind of explain a little more about what the MEA recording is. It's not something that's used by everybody at this point, no electrophysiology is used by everybody, and the MEA is used by even fewer people. So again, you thaw and plate your cardiomyocytes, let them recover for a few days, and then trypsinize them onto these MEA wells. Each section here of this pie chart, this trivial pursuit piece, is an isolated well. This array right here would have an effective N of 6. After 3 to 5 to 8 days, you can put them into the amplifier and record. The beauty of this system is that it's technically less demanding. You don't need a highly trained, expensive employee to sit at a patch clamp all day to get a couple of cells. You can bang through several drugs in a day with this. It really records kind of an extracellular EKG of the cardiomyocyte activity, which is the sum activity of all channels. And it's also a population response. So a lot of the biological variability, cell-to-cell variability, is kind of evened out or dampened out here. So what is the EKG waveform? Well, it's written here, the MEA waveform. It's shown here in blue. And it's really voltage deflections as a function of time. This represents a beat. So each time the massive cardiomyocytes beats, you see this waveform. You've got these initial deflections that are analogous to the sodium and calcium as well as initial potassium currents. This is the QRS complex of the EKG as well as the initial upstroke of the action potential. You then got a plateau phase, which is primarily due to calcium. That's this phase in here as well in the EKG, and the plateau here on the action potential. You then have this T wave, or this secondary hump. I don't want to call it a T wave because it's not an EKG. It's a secondary hump that is analogous to the T wave, and that is the repolarization of the action potential. You got the QT duration that you often measure from the EKG, from here to here, and that's represented from here to here. Any drug that's going to affect either potentially calcium channels or potassium channels are going to affect this difference between here. So you'll apply your drug and you look for changes in either beat rate or QT interval, and that can provide a lot of information about the actions of your drug. So that's kind of illustrated here. Here we've got a massive cardiomyocytes sitting on top of these electrodes. The electrodes are picking up this electrical activity, and that's shown over here. So it's a beat, beat, beat. It's this long string under controlled conditions. This is an aggregate that we've placed down. We typically do these under monolayer conditions. It's much harder to see the beating from a visual perspective as a monolayer, and that's why we've shown you here in aggregates. A typical experiment then is that you start out, and here we just look at the field potential duration as a function of time and drug application using E4031 as our standard drug. We've got baseline conditions under wash conditions. We add 3 nanomolar. Then we start to add 10 nanomolar, and we see an increase in the field potential prolongation, and that is dose-dependent as it shows it even further in 30 and 100 nanomolar. And here we just simply see overlaying field potential durations, which is just a visual representation of that field potential getting longer. Here we looked at 3 other E4 again, but some additional drugs to look at examples of single-channel block. So on the top row here, we see overlaying field potential durations under either wash or control conditions, which is always black, or various levels of drug, which are going to be your other color tracings. So with nifedipine, again, that's a positive force. So if you block that, that's going to shorten the field potential, and we see a dose-dependent shortening here at 10 and 30 nanomolar. And again, this field potential, corrected field potential duration is listed down here on the bar graphs. So we see a significant shortening of the field potential. Under E4031, which is going to block HERG, we see a significant prolongation under the various conditions. And with chromanol 293B, which is going to block IKS, your other repolarizing potassium current, we also see an increase in the field potential duration. So again, with this, looking at single-channel block, we're able to hit depolarizing currents as well as repolarizing currents. Single-channel block is really easy to see, but a lot of drugs are a little sloppier than that, and they'll have a multi-channel block. And to look at that effect, we've chosen a drug flecainide, which has both a sodium channel and a HERG channel liability. If we look at the field potential overlays of increasing concentrations of flecainide, we see a dose-dependent increase in the field potential prolongation. However, when that's corrected for rate effects, we don't see a statistically significant increase in field potential duration. So if you're going to use only this data, you may come to the conclusion that flecainide does not have a HERG liability, but we know that that's not true. So I'm going to really give a high-level explanation of how we look at that. And this is in collaboration with a company out of the UK called Vivo Medica, which is specializing in waveform analysis using principal component or PCA analysis of the waveforms. If you look at this waveform, you can measure the duration from here to here, but you've got a whole lot of other information. You've got the ways that the curve is spread out. So how does the area under the curve look? How does the amplitude of these peaks look? What is the variation from beat to beat? So all of these parameters can be plugged in to really pull a lot more information out of these waveforms. If you look at 2 of these, center of gravity of profile versus the amplitude variation. So the center of gravity is just going to be where can you draw a line that has the equal amounts of the waveform on each side. The amplitude variance is just going to be looking at these peaks. If you compare both of these with E4031 and flecainide, you see that, you do have an E4031-like liability, which would indicate the hERG channel liability. However, if you look at, say, beat rate or the spike amplitude, you see that these two parameters do not match up between E4031 and flecainide, suggesting that there's some other liability going on in there. If you were to plot the principal components onto this 3D axis here, you can see that the data from E4031 is at different concentrations, clusters fairly nicely, versus the data from TTX, which I don't show up here, is clustered very nicely. The data from flecainide, though, however, is spread out over a much larger area. And this is done into 3 distinct groups here. So this long, wide spread out clustering is indicative of additional channels being blocked. So here you've got an assay that's relatively easy to run. You can put your unknown drug on it and then really pull out what channel types or what combination of channels it may be blocking. All right, so that was it for the electrophysiology. Now we're going to get more to cell-based cytotox happening. And again, I just want to show a slide or two here of just introduction to it. Again, you'd receive your iCell cardiomyocytes, take them out of the liquid nitrogen, thaw them and plate them into your 96-well plate. After about 2 to 3 days, you can change the media, begin your drug incubation, and then at the suitable time later, perform the assay. The one thing I wanted to show here is that these are cells that have been plated onto a 96-well plate. This is the entire well being shown, and the cells have been labeled with calcine AM, just to kind of illustrate the coverage that you can get. And that is modulatable depending on your exact needs, but we can get a nice uniform covering from fairly sparse to fairly dense. So it's very easy to manage how many cells down, what kind of coverage you want to have down there. And also, you may wonder, because I'm going to tell you, that the cardiomyocytes, although they're very pure, there's going to still be that 1 to 2% of dividing cells that are present in there. Are those going to take over the cell culture? How do those act under the cell conditions? Well, under just standard media, just DMEM plus serum, that's typical factors, we can actually see a decrease over about 2 weeks, a decrease from near 100% down to about 80% purity of cardiomyocytes. Thus, we've had an increase of about 20% of these proliferating or non-cardiomyocyte cells. However, in the culture media that we're developing at this point, we see that this remains constant. The purity remains constant. So you're able to plate these cells down and then undergo at least a 2-week type of experiment and still have a very highly pure population of cardiomyocytes. So your results are not going to be confounded by interaction with non-cardiomyocyte cells. And here I'm just going to go through 4 separate off-the-shelf assays. This is a multi-tox glow from Promega looking at viability or cell death just in the presence of increasing concentrations of staurosporin. And we see a nice dose-dependent response in both assays, both of these multiplexed assays, with IC50s of around 500 nanomolar. Second is we can get down into more mechanistic types of cell death. Here we're looking at a Cas-based glow, which is indicative of apoptosis in the presence of staurosporin. And we see, again, a nice dose-dependent increase where we've got an IC50 down here of around, again, 500 or so nanomolar. Secondly would be an ATP production, here in the presence of doxazosin. And you see, again, a nice sigmoidal curve with an IC50 down here of around 21 micromolar. And last but not least, we're looking at the mitochondrial membrane potential with the JC10 dye. And here is an indication of 2 curves taken from 2 separate batches. And we see, to the 3rd decimal point at least, identical IC50 values for these. So in summary, I'd just like to say that, as Chris talked about earlier, the iCell cardiomyocytes were manufactured through a controlled industrialized process. This is a process that can give you hundreds of millions of cells that are going to be the same from batch to batch to batch, highly regulated, highly controlled process. We've shown the appropriate cardiac gene expression as well as protein localization and functionality. We've also demonstrated the appropriate application responses, both in ePhys as well as cell-based cytotox assays. And then I think very important here, your application, you can tweak it such that it fits your work week. So you're not having to come in on the weekends all the time. So either you're not doing that work or your employees aren't coming in and griping at you. And last but not least, then the iCell cardiomyocytes, represent an appropriate human-based model for drug discovery and safety profiling. And with that, I thank you, and we'll take any questions. And there has to be at least one before you can leave.
Are those cells, do they show consistent characteristics when you produce them? Are they essentially all the same?
The iPS cells themselves?
Yeah.
So we actually go through a great deal of rigor in terms of analyzing the iPS cells from a starting material. It's been shown in the literature that stem cells, including embryonic stem cells, will start to have a certain amount of drift over time based on passage number. So we're very cognizant of monitoring karyotype as well as expression of a variety of different genes in those iPS populations, and then making sure that through the manufacturing process we keep them within a window of regulated passage numbers, such that we're at an early stage, so that we don't see that drift, and basically freeze them back and maintain them for the manufacturing process. So we're only looking at, maybe 10 passages that we use for the entire manufacturing process.
Getting into something a little bit different now, can you quantitate the proliferation potential of those cells?
Yeah, the doubling time, you mean? Or the pluripotency?
No, the pluripotency. The stemness or the primitiveness of those cells.
That's actually an active, ongoing research field to really understand, because the classic methodology for understanding pluripotency is both the characterization in a variety of different differentiation protocols to make sure that they go into all four lineages: mesoderm, ectoderm, endoderm, and germ. But then also to look to see whether if they're put into a immune-compromised mouse, whether they produce a teratoma. Now, that's been the classic pluripotent measure. Really, what the field, including Dr. Yamanaka in Japan, is looking at are there other measurable characteristics, and can there be a toolbox of assays developed to really understand pluripotency without having to go to a full teratoma-type assay? So I would say that we're still in the early days of that field, that research. But from our perspective, when we look at using ES cells as a quote-unquote gold standard, although I think most people would agree that you can't really use it as that, we haven't seen any differences in terms of the differentiation potential of these cells compared to embryonic stem cells. So a lot of information to answer a simple question, but it's an ongoing area of research, and it will be one that will be very important as this technology moves towards autologous therapeutic applications, to really understand both the starting material and any cell types that are derived therefrom.
What's the percentage of the ventricle versus the atrial cardiomyocytes?
We typically see around 50% ventricle, and then the rest, the remaining 50% is divided then between the atrial and the nodal leg in roughly equal proportions.
Okay, now, and also based on the kind of EKG waveform that you recorded using the microarray technology,
Yep.
And you don't see the P wave, so I was wondering if you increase the percentage of the atrial cardiomyocytes, will you be able to detect that wave also?
You may be able to, but I think that's really pushing the analogy between an MEA waveform and an EKG a little too far, because obviously in the heart you've got a clear separation between the atria and the ventricle. In the MEA, the monolayers that are used to record from the MEA, you've got the cells pretty much all together, so you don't have that physical separation to look at a specific atrial depolarization versus ventricular depolarization. So I guess the short answer is no, I kind of doubt it.
Can we specify kind of the different percentage of the mix of those cardiomyocytes?
No, at this point we can't. However, we are developing, as Chris kind of alluded to, putting in some inducible-driven type of promoters. And so that's an upcoming version where we can have your pan-specific cardiac promoter, as well as other inducible promoters that may drive the differentiation or allow you to select for a specific cell type.
Right.
But currently, we've got a pan-cardiac population.
Yeah, the first step is pan-cardiac to give a full kind of in vivo-like response, and then really relatively simply through the genetic engineering of the iPS cells, we could do an atrial-specific promoter or a ventricular-specific promoter and create those populations.
As soon as we can make it.
As soon as you buy a lot of pan-cardiac.
Just a general question. This is my first foray into learning about the iPS cells. Mm-hmm. And you had mentioned that you use a special recipe of cytokines and growth factors to cause them to differentiate into cardiomyocytes. And I'm assuming that your cellular dynamics special medium has a certain recipe of these same...
Like use secret sauce.
Same growth factors and cytokines. So when you mention that if you don't use that special medium, there's possibly 1 to 2% of non-cardiomyocytes that might be taking over that dish, is it also possible that your cardiomyocytes are reverting back?
I doubt it. We haven't particularly looked at that, but we don't see any measurable levels of, say, the stem cell genes. But that wouldn't be necessary for it to go back. No, you'd be asking if we got a cardiomyocyte. I mean, I'm trying to think of this. I think kind of the analogous situation would be where if you took did a dissection of a primary skeletal muscle and put it into culture and it starts to de-differentiate. We really haven't seen any type of upregulation of those type of stem genes or any other type of transcription factors that would make that. So I think that's fairly unlikely.
The hope is that one will be able to then transplant these back into the individual you took them from, and they're mostly going to be elderly, aging people, but your mitochondria are old. So the mitochondria from these cells has accumulated, there's a lot of heteroplasmy, you've accumulated mutations, and mitochondria from old rats are quite different from mitochondria from young rats. So that's a problem, though I think there are ways of getting around that.
Yeah, that's also primarily why we're not focused on the therapeutic application of these cells today. I think there's also a number of other hurdles that will be required to be overcome in order to, at least for cardiomyocytes, from a cell therapy perspective. And it's really one of the reasons why the company is focused on the strategy of providing research tools initially and to develop this manufacturing process. The others are based on engraftability and cell delivery to the organ of interest, right? So many of these pathologies, you mentioned the mitochondrial function, so you first have to get the cell to the right organ in general, right? So that's a challenge. The second thing is just by replacing cardiomyocytes into an organ that has congestive heart failure, for instance, does not fix the pathology that generated the congestive heart failure to begin with. That was due to lack of blood flow. So just putting in more heart cells is not going to fix that problem. And then in addition to that, you have, as we talked about the other day, that these cells could revert and potentially perform cancers in the individual. Those are all hurdles that are going to need to be overcome to make these types of cell therapies a reality. And we feel that and Dr. Thompson has spoken on this publicly, that we believe that there's going to be way more effective, better therapies developed from the use of stem cell-based human models than there will be cell therapies developed from stem cells. And we run the risk of kind of taking in the hype of what the potential is without the appropriate guarded optimism that's necessary to get there. So Dr. Thompson always puts a paragraph up about talking about gene therapy and the downfall of the use of that as a therapeutic application, and basically says, look at this paragraph, and everywhere it says gene therapy, you could say stem cell therapy, and we could run the same risks of that problem. And so from the standpoint of our company, that's why we've really focused on providing better cellular models for the existing and current drug discovery process.
We do a lot with primary human cells in culture and making them live longer. And old cells don't live as long as young cells. And one trick is everybody's growing them in the wrong way. They grow them in air, and you should be growing them in 5% oxygen, because that's the concentration of oxygen in the body. So if you're growing your cells in air, you're giving them a huge oxidative stress. Anyway, there are lots of tricks, I think, that can be done.
Yeah, agreed. I mean, another one is that just the three-dimensional structure of the cells. I mean, we can put these in potentially an environment that puts the populations of cells in a more in vivo-like environment. But really, I mean, the trick up to this point was, in terms of old versus new or primary, is really has always been the batch-to-batch variability, right? In terms of you can only get a limited quantity, they have limited proliferative capabilities, and in order to do longitudinal studies, they're really not that useful as they could be. And so by being able to produce a highly homogeneous, significant population of cells that are consistent from batch to batch, it potentially provides a higher utility of tool compared to primary cultures.
Coming back to growing cells under low oxygen tension, I've been doing that since 1982, and all of our studies that we do at Hemogenics are all performed under low oxygen tension.
Yeah, one of the other things that we're looking at, just in terms of cardio function, is we're putting these cells into a low glucose environment. Most cell culture medias have a physiological concentration of glucose. So if you're looking for drug effects that affect mitochondrial function, having them in high glucose, you basically are never stressing the mitochondria, so you basically will never see that phenotype. And so one of the things that we're doing with these cells is putting them in a low glucose environment such that we can manifest that mitochondrial function.
I have a few questions. First question is about the quality of the cells. So I'm just wondering whether if you have any criteria in terms of a volunteer selection, such as like age or something, is there a limit?
Well, the first step in terms of developing is the appropriate informed consent. And that has been signed off on as to how these cells are going to be used. We're looking at a variety of different characteristics. Are you talking primarily about the ethnic diversity type of approach, or just any iPS cell? We want to represent a clinical population that is useful for the pharmaceutical environment and the populations that you market your drugs to. So we will look at all demographics of an individual and not really apply any criteria per se, but have the ability to both try to represent a representative population from a panel perspective, as well as have the ability to do custom iPS derivation from samples that a particular partner would find useful.
Actually, the reason I'm asking the question is because I imagine, a different individual may respond, the same treatment differently.
Absolutely.
Okay. The second question, that for now, each batch, is it from each individual or is it a mixed population?
Right now, the first product will be from a cell line derived fibroblast, commercially available. Okay. So these are just a generic cardiomyocyte, and we're in the process of building out the iPS panels right now from a variety of different individuals. So those will be kind of in the next wave of products.
In the next wave. Okay. In terms of that, I'm just curious, see if you have any idea. Say you get a cell from like a 70-year-old or a 20-year-old. In terms of their cardiomyocyte, would that be any difference? Do they mimic kind of the age from the donor, or they are kind of, even the older one, their cardiomyocyte from the iPS is still kind of a younger without all the whatever.
We haven't been able to do those studies yet. The only thing I could say is that we've been able to derive cardiomyocytes from a large continuum of ages of samples. So the oldest was a 92-year-old individual that we were able to make iPS cells from and then generate cardiomyocytes. I think at this point it's difficult to ascertain whether that phenotype, based on the current measures, is representing that individual's phenotype. One of the things that is the key focus of this area is to look at genetic predisposition or genetic diseases, which is why Dr. January and Dr. Camp have initiated a grant to be able to produce iPS cells from people that have genetic long QT syndrome, for instance, such that we can represent as a potential positive control for long QT prolongation or torsades.
Okay, thanks.
Any other questions? Great discussion. Thanks a lot for your attention and attendance here today. Please feel free if you want to continue the discussion or think of anything else that you want to talk about and stop by the booth. But we certainly appreciate your attendance. Thanks.